This is the OncoZine Brief with Peter Hofland and Sonia Portillo.
In this episode of the OncoZine Brief, we talk to Sean McCarthy, President and Chief Executive Officer at CytomX. Dr. McCarty joined CytomX in 2010 and has more than 20 years of experience in the biotechnology industry, which includes roles in R&D, business development, financing, and general management. I'm Peter Hofland, here with Sonia Portillo, and this is the OncoZine Brief.
Dr. McCarty held research leadership and program management roles at Millennium Pharmaceuticals, where he managed biologics discovery programs, SGX Pharmaceuticals, where he spearheaded a wide range of large pharma partnerships as Vice President Business Development prior to the acquisition of the company by Lilly, and was a transactional partner at venture capital firm Pappas Ventures, responsible for investments in therapeutic, medical device, and molecular diagnostic companies, including Afferent Pharmaceuticals, which was later acquired by Merck, Tyrex, acquired by Medtronic, Lumina, acquired by Shire, CoLucid, acquired by Lilly, and Lead Therapeutics, the originator of talazoparib, now a Phase 3 product candidate in the Pfizer pipeline. Dr. McCarthy is also an author on multiple peer-reviewed scientific publications, issued patents, and filed patent applications.
CytomX is unlocking the potential of antibody therapeutics in oncology by developing a novel therapeutic antibody class of highly targeted so-called Probody Therapeutics.
In our interview with Sean McCarty, we talk about the history of CytomX, the development of a new class of highly targeted drugs, basic research, and the company's collaboration with industry partners. Let's listen. Dr. McCarthy, thank you for joining us. So first off, can you explain CytomX's Probody Platform and how is this different from antibody-drug conjugates?
Yes, well, pleasure to be here today and to have the opportunity to tell you something about our company. First of all, let me just tell you at a high level what CytomX is and what we're trying to do. We're a clinical stage biopharmaceutical company based in the San Francisco Bay Area. We are developing a deep and differentiated pipeline of oncology therapeutics based around what we consider to be a very innovative approach to therapeutic antibodies that we call Probodies. Probodies are different from typical conventional therapeutic antibodies in one very important way, which is that we add what we call a mask to the antibody, which is designed to prevent the therapeutic antibody from engaging with its target until the mask is removed. And the mask is removed very specifically within cancer tissue by proteins in cancer tissue called proteases. So the net result of this approach, this Probody approach, is that we can localize or concentrate the therapeutic antibody within cancer tissue and spare binding to normal, healthy tissues. And this allows us to do a couple of things. First of all, to broaden what we call the therapeutic window for antibodies, which is the balance between activity and toxicity. So improve therapeutic window where there's room for improvement. But we can also create a therapeutic window where one did not exist previously for very high potential oncology targets that have not been able to be addressed before. And this is allowing us to create a pipeline, a deep pipeline of clinical programs, which are either best in class or first in class. And by the end of this year, end of 2018, we plan to have 5 programs in clinical trials.
So if you look at this technology, the drugs that you are developing, it sounds that there are some similarities with antibody-drug conjugates, but there is also a big difference between the drug that you develop, the Probody antibody technology, as well as the antibody-drug technologies. Can you really summarize some of that difference?
Absolutely. And I would say that the technology itself applies really to all therapeutic antibody formats, of which the antibody-drug conjugate is just one. So the underlying technology, the masking technology that we have developed, we have applied to therapeutic antibodies, to antibody-drug conjugates, to bispecific antibodies, and we're also researching the application of masking to certain CAR T therapies, which are, of course, engineered immune cells engineered to present and recognize tumor cells in a very effective and potent way. So the underlying technology itself is applicable to really all antibody formats, and not specifically antibody-drug conjugates. And I can tell you more about each of those in terms of the programs that we have in the clinic. For example, the lead program in our pipeline is a Probody against PD-L1, which is a very well-known, very well-validated target in oncology. So this particular therapeutic is an antibody against PD-L1 that we developed, which you can think of as somewhat similar to the antibody that Roche is commercializing, which is called Tecentriq. This is an anti-PD-L1 antibody. It's a very effective anti-cancer therapy in a number of indications. We have our own anti-PD-L1 antibody, but we have put a mask on it, and the mask allows us to focus the activity within cancer tissue and diminish the side effects. So that's our lead program, which I can tell you a bit more about later on. The second program we have in the clinic is what we call a Probody drug conjugate. So now this gets to the antibody drug conjugate application. This is a very interesting molecule that is, again, it's an antibody against a very abundant tumor protein we call CD166. This is a molecule that's present on the cell surface of many different cancer cells in many different cancer types. We have an antibody against CD166. We turned it into a Probody by putting a mask on it, and then we conjugated it to the payload, as you would a typical antibody-drug conjugate, to make the Probody drug conjugate. And the reason that we've done this is that this target, CD166, we believe has great potential as an anti-cancer therapy. The problem is that this target is also present on many normal human tissues. So no one has tried to develop a drug against this target before because it's on normal tissues and normal cells, and it would be predicted to be highly toxic in patients. The Probody approach allows us to selectively target in tumor tissue and not target or bind to target in normal tissue, and in doing so, open a therapeutic window for this very high potential target. So that's just another example of an application of our technology. And fundamentally, what we're trying to do with our platform technology is to generate a whole new class of safer, more effective anti-cancer therapies. And that indeed is our vision statement for our company, which is transforming lives with safer, more effective therapies.
Tell us about your IND for CTLA4 Probody therapeutic, and what makes this drug unique, and how does it address an unmet medical need?
Yes, this is a very exciting program that we're moving forward with our partner, bristol-myers-squibb. And this goes back to a foundational alliance we formed with BMS in 2014. This molecule is a Probody targeting an immune checkpoint called CTLA4. CTLA4 is the target of the BMS drug Yervoy, which they launched in 2011, and which was a foundational advance in the field of cancer immunotherapy. In fact, it's the drug that really ignited the whole field of cancer immunotherapy several years ago. This is a really Yervoy, which is the BMS drug, is a very powerful drug. It causes or results in complete remissions in certain cancer patients. However, it's also a rather toxic drug because its mechanism of action is to activate the immune system to recognize cancer cells. Unfortunately, the activated immune system also can recognize and damage many normal tissues as well. So what we're trying to do with BMS, with the Probody, is to develop a safer, more effective version of Yervoy. So we did that by applying our masking technology to Yervoy itself, putting a mask on the antibody, a mask that can be removed in tumor tissue by tumor-associated proteases,
And BMS advanced that molecule into the clinic recently, and they're currently running a phase 1/2 trial evaluating the CTLA4 Probody, which they refer to as BMS 986249. They're evaluating it in a range of tumor types, both as a monotherapy and in combination with an anti-PD-1 inhibitor called nivolumab. So we're very excited about this program, and it's the leading edge, as I said, of this foundational alliance that we have with BMS, and really is a great illustration of the potential power of our technology to create safer, more effective medicines for cancer patients.
I see, and you're also developing a new drug called CX7 What is this being investigated for, and what are some of the results that you've seen thus far in preclinical studies, and were they as what was expected?
Yes, this is another very exciting program. This is a program that we still own full rights to at CytomX. It's our most advanced molecule in clinical studies. It's a Probody that targets a protein called PD-L1, which, like CTLA4, is another immune checkpoint target. And similarly to anti-CTLA4 antibodies directed against PD-L1 have been shown to be very effective in a range of different cancer types in inducing potent anti-cancer responses. Similarly to CTLA4 inhibition, blocking PD-L1 also can result in severe immune toxicities. And so the approach here is very similar to the approach we're taking with BMS on CTLA4, which is to make a masked antibody, a Probody, against PD-L1 with the promise of a safer, more effective version of the drug that can be, again, dosed optimally in patients, and in particular, that can be dosed more optimally in combination with other anti-cancer agents. The way the field of immune therapy has evolved over the last 5 years, it's really become a field of combination therapies where there are many, many clinical trials now underway evaluating the combination of PD-L1 inhibitors in combination with other anti-cancer agents. And the reason for that is that although the activity of PD-L1 as monotherapy is pretty good, we know it can be improved by combining with other mechanisms. As you might imagine, however, when you combine multiple drugs together, you, in addition to seeing increased activity or efficacy of the combinations, unfortunately, we also typically will see increased toxicities as well. So the vision that we have for CX7, the anti-PD-L1 Probody, is that it could become a safer and more effective centerpiece of combination cancer immunotherapy moving forward.
Does that mean that you are you can now manage these the adverse events that you may see in an uptake if you combine different drugs, but actually reduce the effects of them because you are actually masking one of the drug's activities?
That's exactly right, and that's exactly what we're evaluating in our clinical trial that we call PROCLAIM CX7. PROCLAIM stands for Probody Clinical Assessment in Man. This is a broad-based phase 1/2 clinical trial that we launched to evaluate the safety and activity of CX7 as a monotherapy, but also in combination. And we have 2 combination arms in this study underway. One is with Yervoy, the BMS drug that I just mentioned, and the other is with a small molecule called Zelboraf, which is an inhibitor of the protein kinase BRAF. Now, we know from prior published data that combinations of PD-L1 or PD-1 inhibitors with either Yervoy or Zelboraf are significantly more effective as anti-cancer treatments than either agent alone. So the combinations clearly are more active, but the combinations are also considerably more toxic. Indeed, the synergy of activity is mirrored by the synergy of toxicity. So the masking of the PD-L1 Probody is aimed at improving the tolerability of the combination And the rationale for that is that in antibody form, the PD-L1 antibody activates the immune system broadly, but in Probody form will only activate the immune system in the tumor. So that's what we're evaluating in this clinical trial, and we'll present our first data in the middle of this year. If you just think about CX7 as a molecule, the anti-PD-L1 Probody, it's really taking advantage of two fundamental biological observations that we have learned about cancer. The PD-L1 component takes advantage of the emerging understanding of checkpoint molecules and how they control the immune system, and how if we block the checkpoints, we can reawaken the immune system to recognize cancer tissue and eliminate it. The molecule also takes advantage of many decades of research into how cancer is rich in these proteins we call proteases, which play an important role in the formation and metastasis and spreading of cancer. We're taking advantage of these proteases to activate the anti-PD-L1 Probody. So this is a molecule that really builds on decades of scientific research and tries to turn these properties of cancer tissue back on the cancer itself by taking advantage of these fundamental intrinsic biologic mechanisms to create a very, very innovative and we hope ultimately very effective anti-cancer agent.
So if you look at the whole series of products that you've been dealing with, I mean, it's changing the way we actually deal with cancer or how we treat cancer. It's potentially creating a safer environment or safer tool for oncologists to work with patients. What are some of the things that really we need to look at in the future to change a little bit further? It seems to me a very exciting development that you're dealing with.
Yes, absolutely. It's a very good question, and in fact, the second program that's in the clinic at CytomX, which we call CX2009, I think is a very good example, a very good illustration of the potential for Probody technology to bring therapies to patients that otherwise could not be developed. And this particular molecule, again, CX2009, is what we call a Probody drug conjugate. This is a first-in-class molecule that is directed against a very abundant tumor antigen called CD166. This is a protein that's on most tumor cells, most tumor types. And it is, we believe, a terrific target. The challenge with the target is that it hasn't been developed before because this target is also on many normal tissues. And you would predict that if you made an antibody, or particularly if you made an antibody drug conjugate to CD166, you would predict that it would be highly toxic in normal tissues. So no one has taken a CD166 molecule into the clinic yet.
That toxicity is caused by the fact that you have the same target on so-called healthy cells as well as on cancer cells?
That's right. And so for a therapeutic without some kind of masking or some kind of way to localize in cancer tissue, a therapeutic against CD166 you would predict would be toxic to patients and would have what we call would not have a therapeutic window. So what we've been able to demonstrate in our preclinical studies is we've made a Probody to CD166. We have conjugated it to a highly toxic payload that is called DM4. That's a molecule that we licensed from a company called Immunogen. This is a very potent molecule that can kill cancer cells very effectively when the mask is removed, but that can't bind normal tissue because in normal tissue the mask stays on and blocks the antibody from binding to its target. So the Probody approach here is allowing us to develop a first-in-class therapy with potential across many different cancer types, against a target that no one else has been able to take forward into the clinic before. And I think this is a terrific illustration of the true power of our technology to bring new medicines to patients that would otherwise not be possible.
Right. So this whole area is within oncology, and that's often an area of expertise where a lot of the more advanced drugs, the more difficult drugs to treat disease with, are being tested or being developed. The technology comes out of that. If we look right now at ADCs in general, and we look at your technology, the so-called PDCs, if I may call them that way, what are your expectations to take this outside of the realm of oncology, so beyond oncology and hematology, so to speak?
Well, when we think about the Probody platform as a whole, of course, the technology platform is based upon the scientific observation that these proteins called proteases are switched on in cancer tissue and switched off in normal tissue. And that's something we've known for a long time. It turns out that proteases are also turned on in a number of other diseases. In fact, many different diseases have active proteases: inflammatory disease, cardiovascular disease, tissue remodeling, tissue fibrosis. There are many additional indications beyond cancer where proteases are switched on. And it's certainly true that all of these could be opportunities for Probodies, potentially for Probody drug conjugates in some form. Interestingly, there has been some work done in the field, not by CytomX, but by others, looking at very creative antibody drug conjugates in inflammatory disease, where the conjugated drug put on the antibody is not a cytotoxic molecule that's meant to kill cells, but is something that looks more like an anti-inflammatory, for example, a steroid. And the idea being that you could more locally deliver with an antibody, you could deliver anti-inflammatory therapy. And those kinds of approaches, we think are interesting, and could lead the way to the ability for CytomX to build programs outside of oncology as we continue to build and develop the company. So the technology has, we think, very broad application, and it's worth noting that in the Bristol-Myers alliance that we expanded last year, we announced a major expansion of our alliance with BMS. They paid us an additional $200 million up front to gain access to 8 additional targets from our platform. And 2 of those targets will be outside of oncology. So BMS has certainly recognized the potential for our technology outside of oncology as well. And we will continue to think about, as we grow and develop our company, about making investments in non-oncology areas as well.
So one of the things that comes to mind if you look at antibody drug conjugates, and not necessarily your technology, but that's just dealing with antibody drug conjugates, is that if you may switch to some areas outside of oncology, one of the problems that you may see is an effect in cells that not necessarily be targeted. The so-called off-targeted response that you may see. Is that something you have also with your technology? My understanding is talking to some of the people at Merck, for example, that they really came across this problem and had to change, for example, the linker technology between the cytotoxic and the actual antibody. In this case, is that some of the things that you come across in your technology as well?
Yes, absolutely. And if we take lessons from the application of antibody-drug conjugates in oncology, some of the key lessons have been that the target itself that the antibody engages with, the target needs to be present at high levels in the disease tissue to enable enough of the antibody-drug conjugate to bind to the cell and get inside the cell and actually ultimately kill the cell. So we now have a pretty good understanding that we need targets for ADCs that are present at high abundance in cancer tissue. And in fact, if you look at the most advanced, best known antibody-drug conjugate that is Kadcyla, the Genentech Roche drug that targets HER2, that drug is effective in breast cancer because HER2 is present at high levels on many breast tumor types. And so we know that high level of target is required for activity. HER2 is not expressed at any significant level in any other tumor type, and therefore Kadcyla does not work in any other tumor type. So taking that learning, if we ask the question, which proteins, which targets are present at the highest possible levels in cancer tissue to give us the highest chance of getting enough of the toxin into tumor cells, it turns out there are many targets that are present at even higher levels than HER2 on many different cancer types, but they have not been developed before because those targets usually, almost invariably, are present on many normal tissues, like CD166, and another target that we like CD71. So if we then move outside of oncology to think about other indications, I think we have reason to believe the same rules should apply, that you would want targets that are present at high levels in the disease tissue. And what we're able to do with our technology is, regardless of whether those targets are present on normal tissue, we're able to develop drugs against them with a therapeutic window because the Probody masks the binding outside of the disease tissue. So we have, as I said, we have reason to believe that the same rules might apply outside of oncology as well. Additional research is required.
After the break, we're back with our interview with Sean McCarthy, President and Chief Executive Officer at CytomX. And welcome back to the OncoZine Brief. I'm Peter Hofland, here with Sonia Portillo. Today we're talking with Sean McCarthy, President and Chief Executive Officer at CytomX. So you were mentioning a little bit to close the program with about your relationship or your partnership with Bristol-Myers. What kind of other organizations and companies you're working with in the development of your Probody technology?
Yes, well, partnering has been an important part of our business model for several years. And we believe fundamentally that to grow and develop a platform technology company and turn that company into an integrated biotechnology company, partnership formation is a very important part of the overall business plan. So to date, we have formed alliances with 4 major pharma companies, with Pfizer, with BMS, with AbbVie, and most recently with Amgen. The deals with Pfizer and AbbVie are focused in the Probody drug conjugate space. The deal with BMS is in oncology, including immuno-oncology. And our most recent alliance with Amgen is a very interesting deal focused in an application of Probody technology in the emerging field of T-cell engaging bispecific antibodies, which is a very exciting emerging format of therapeutic antibody that we believe has very, very high potential, and Amgen believes as well. Let me just tell you just a little bit about what these molecules are. So in contrast to an antibody-drug conjugate, where you have an antibody with effectively a chemotherapy on it, the T-cell engaging bispecific does something a little different. It's a bispecific antibody that can bind to cancer cells, but by also binding to T cells, it can recruit T cells into cancer tissue, and those T cells can attack the cancer cells and ultimately kill them. The first approved T-cell engaging bispecific is a drug called Blincyto, which was initially developed by Micromet. Micromet was acquired by Amgen several years ago, and that was a real breakthrough in the field. And I would say across the biopharmaceutical industry in oncology today, there's enormous interest in finding new T-cell engaging bispecifics. As you might imagine, one of the challenges is, just like with immunotherapy, if you have a bispecific antibody that can activate T cells, these molecules can be very, very they're very potent, and they can be very toxic because, again, you activate T cells throughout the entire body. So what we've been able to show preclinically with Probody technology applied to these T cell bispecifics is that, again, as with our other programs, we've been able to substantially improve the therapeutic window, the balance between safety and toxicity. And we've presented some of this work at a number of scientific meetings. And Amgen, as the leader in the field since their Micromet acquisition, was very excited in our preclinical work. And we were able to sign this deal towards the end of last year. And we're now moving multiple programs through early stages of R&D, and we're absolutely thrilled to have Amgen as a partner.
This is very exciting news, very exciting to see that you where you're going with this technology. When can we expect the first Probody-based drugs in the clinic, in not in clinical trials, but actually for treatment options?
Yes. So as I mentioned, we have 2 Probodies in clinical trials ourselves. That's CX7 and CX2009. BMS has the anti-CTLA4 Probody in clinical trials, and by the end of this year, we'll have 2 additional programs in the clinic. One of those will be what we call another Probody drug conjugate against a protein called CD71. And the 5th program is an anti-PD-1 Probody. So we will be presenting our first clinical results this year, in middle and second half of 2018. And of course, depending upon those initial data, we'll be in a position to think about what the next stage of clinical development might look like. And it's too early to guide on precisely when Probody Therapeutics could be available for patients, but this is an important year for us as we will get the first initial views as to how Probody technology is functioning in patients.
Definitely exciting. Well, thank you very much for being here with us today, and we definitely will be coming back with future interviews with you. I'm sure about that.
Great, it's been a pleasure. Thank you very much.
This edition of the OncoZine Brief was originally recorded on February 9th, 2018. For us here at the OncoZine Brief, we want to thank you, our listeners and underwriters, for your ongoing support. Thanks to your support, our program now has a wider reach. Our program can be heard in Arizona on KFNX, one of the top 10 radio stations in Arizona, reaching almost 5 million people throughout the state, as well as via iHeartRadio in addition to PRX, Public Radio Exchange. And you can also download our program via iTunes. Later this month, UK Health Radio will also start broadcasting our program in the United Kingdom and mainland Europe. For more information about that, check our online journal, OncoZine, at OncoZine.com.
We know that based on this interview, you may have questions, so please submit your questions to our editorial team via email, Facebook, or Twitter. We will post as many answers as we can on our website, OncoZine.com. That is OncoZine.com. Thank you all, and thank you for listening. And join us again for our next episode. I'm Peter Hofland, here with Sonia Portillo, and this is the OncoZine Brief.
The OncoZine Brief is produced for Sun Valley Communication by Peter Hofland, Sonia Portillo, Evan Wendt, David Kaler, and Sean Mayer, and distributed by InPress Media Group. Support for the OncoZine Brief comes from listeners of this station and our commercial underwriters and advertisers. For more information about underwriting and sponsoring options, contact Sean Mayer in California at 949-923-1660 or visit our website at OncoZine.com/underwriting. The OncoZine Brief contains health and medicine-related information and is provided for educational and entertainment purposes only. The content is not intended as a substitute for professional medical or health advice and does not replace your doctor's advice. Your doctor is the best person to answer questions about your personal health. If you hear something in this program that doesn't agree with what your doctor has told you, ask him or her about it.
In the next episode of the OncoZine Brief, we talk with Dr. Gisela Swab, President, Product Development and Medical Affairs and Chief Medical Officer at Exelixis. Founded in 1994, Exelixis is an oncology-focused biotechnology company that strives to accelerate the discovery and development of new medicines for difficult-to-treat cancers. Following early work in model genetic systems, Exelixis established a broad drug discovery and development platform that has served as the foundation for the company's continued efforts to bring new cancer therapies to patients in need. The company discovered and developed its lead compounds, cabozantinib and cobimetinib, and advanced them into clinical development before entering into partnerships with leading biopharmaceutical companies in an effort to bring these medicines to patients globally. Until next time, this is the OncoZine Brief.